Okay, so I told you about the different kinds of light microscopes, and now we need to talk about electron microscopes. There's a huge difference between a light microscope and an electron microscope, but first let's talk about what we gained from creating an electron microscope. We can see bacteria, and you're going to do that tomorrow when you gram stain. We can see bacteria under a light microscope. What can't we see? We can't see viruses under a light microscope, and we really can't examine DNA. So anytime you want to get to internal structures of the cell or see viruses, you have to go to an electron microscope. So I'll hesitate here for a minute and let you write. So in that picture, that's an electron microscope. She may have eyepieces there for getting it into focus in general. Once she switches to the electron power, then she looks at the computer screen. Because basically what happens is with an electron microscope is this. There's a special preparation you do for your sample, and I'll talk a little bit more about that a little bit later. But then you shoot electrons at the specimen. The electrons bounce off of the organism and are recorded using a screen that kind of lights up where the electron hits. All of that is hooked to a computer, and the computer takes all that information and creates an image. The electrons don't care what color the specimen is, so any color that you see in an electron image is added for contrast so you can see things better, or for labeling purposes so you can tell what's the bacteria versus the medium it's being grown in. So these use a beam of electron, not light. So then there's no real reason for lenses, glass lenses. So they're kind of like mirrors. They're called electromagnetic lenses. And that kind of helps the electrons bounce at the specimen from different angles, so you can get more of a three-dimensional image. Okay, hopefully that was enough time to write. I'm going to switch the slide. So the special preparation I was talking about, at least one of those is called shadow casting. I'm going to let you write and then I'll explain it. So about the best description I could give you of this is it's kind of like spray painting something, and the paint is made of metal. So you spray the organism with this metal, and the reason that helps is because the electrons bounce off of metal better. So again, it helps a lot with shadowing and that kind of thing. Across the top of this slide are electron images. The one on the left they have not enhanced with colors. The next three they have, and they did that to highlight the bacteria versus whatever it's growing in or on. So they give it a little contrast so it stand out better. So there's two main ones that we use in microbiology, and then there's a couple others I thought I would mention because they do have some significance to genetics and biology. So there's many different kinds of electron microscopes. These four are not the only ones, but I had to draw the line somewhere at what you should know, so I drew it here with four. The first one is actually the most powerful one of the four, and I'll let you write that before I explain. The transmission electron microscope is used, it's like I said, it's got the most powerful magnification of the four that I have listed here. So it's if you need to go deep and see internal structures, the nuclei, the organelles, etc. It magnifies anywhere from worst case scenario 10,000 up to best case scenario 100,000 times. It's also the most expensive of the different microscopes. Then there's the scanning electron microscope. This one doesn't magnify as much, but it's also not as expensive. I'll let you write and then I'll explain. The outside of a bacterial cell is the cell wall, and on that cell wall it's pretty much made of proteins, some lipids, that kind of thing. But on that cell wall are receptor sites. The cell needs those to take in nutrients and give off waste. But sometimes and human cells have these too. So sometimes on the cell, parasites, either bacteria or viruses or something, will attach at those receptor sites if they're the right shape to fit there. So a scanning electron microscope is really good for examining those receptor sites and the shapes, which can lead to finding ways to keep things from attaching there that don't belong there. So that gives you an idea of the scanning electron. Now, again, it's not as powerful, so you're not going to see as detailed internal structure as you would with a transmission. And then two more. The first one is the scanning tunneling microscope. And I'll let you write. The scanning tunneling microscope is used from our point of view, to actually examine DNA. It can also be used in, well, a lot of different things, but also to examine the silicon chips in the computer's motherboards. So they can actually do repairs to something that's not even visible. So I don't claim to be an expert on that particular aspect, because that's something that I wish I had more knowledge in than I do. So, and in the computer world is constantly changing, so it's real hard to keep up with what's going on with that. But anyway, the scanning tunneling microscope, they are using to work with some of the more intricate stuff in the computer hardware. And then the last one is atomic force microscope. This one, they can actually examine enzymes and see how they work and what stops them from working. Enzymes, we know, are very sensitive to temperature change, and so we didn't know why. Well, once they examined the enzymes action under a microscope, they found that at a higher temperature than what they're built to function at, the molecule actually unwinds itself. So the enzyme basically comes apart, and it's called denaturing the enzyme. Denature. Anyway, we'll get to that, but the atomic force microscope is very useful for some of those biological molecules like proteins, enzymes, lipids, and understanding how they are built on a molecular level. And that information is very useful in how to destroy them, repair them, or connect to them. So, anyway,